we just welcome our speakers, Sten Linnarsson and Deanne Taylor. Sten, are you ready to present?
Yes. I will switch to the slides. I hope you can see my slides now. So I want to thank Aviv and Sarah and all of the other organizers here, and thanks for the great introduction. You covered a lot of ground there, so I'm going to keep this short in the interest of time and just make a couple of points. And I'll end on a what's missing slide, where I'm going to try to set out the direction of what we need to do more and better as we go forward. But first, I wanted to start with this slide that goes back to the justification of why we're doing this, why are we making a developmental cell atlas. And of course, there can be many reasons, but for me, a developmental atlas is really meant to be a reference for studying human disease. And in this slide, I'm showing the example of the brain development and brain disorders, and it's very obvious. I think about a third of all brain disorders have a developmental origin, and even those that occur at in late adulthood, like most brain tumors and degenerative diseases, those diseases often show features of developmental processes that kind of recur. That's very clear in glioblastoma, for example. So the study of brain development and more generally human organ development in the healthy human should really serve as a reference. So what do we need out of such a reference? Well, we need ways of anchoring our disease states, our disease data. So we need a coordinate framework, an anatomical framework that doesn't really contain just one time point, but spans the whole time span of human development, so that we can align patient samples onto a particular space and time coordinate. And then we need cell states, which you can get from single-cell RNA-seq data, for example, so we can compare our diseased cells with the healthy cells. And ideally, we need true developmental hierarchies, so lineages, cell trajectories, and so on, so that we can study cell of origin in cancer, or we can study the recurrence of developmental pathways in adult disease. As Sarah mentioned, there are particular challenges in human development. Particularly, it's very difficult to access the earlier stages post-implantation, and she showed some model systems. There are some great model systems. There is a period that is relatively easy to access. It differs in different countries. In my country, it's easier to get access to first trimester. In the US, I think second trimester is sometimes easier. But the third trimester is really difficult everywhere, obviously, for obvious reasons. So again, we will need to rely on model systems, and we shouldn't forget then to leverage all the great model organisms that have been studied for such a long time. And we should try to go in tandem with human development and the development of model organisms. And really, some of the most powerful work to date has been done in model organisms. For example, this C. elegans atlas that really almost captures the entire development of that animal in a single picture. And likewise, earlier work from Alex Shear and Aviv Regev's labs in mapping zebrafish development, and again showing these beautiful lineages that really give a glimpse into what we should aim for in human atlases. We just published this in mouse. Sarah showed it. I just want to point out that here we try to really integrate these various aspects of development that are so important. So time, cell state, lineage, and spatial information. So you can see this almost as an actual atlas where you travel from the early embryo and you sort of travel along the neural tube, maturing into neurons on the right here, into astrocytes and oligodendrocytes on the top, and even into the meninges here on the left. And then you have all the particular cell states around the periphery here. So I think this is kind of the sort of integration that we should aim for also in the human. But I think what's been understudied in all organisms, but in particular in humans so far, are the spatial aspects, and that's coming along now. It's catching up. The technologies are improving. It's beginning to be possible to use, for example, single molecule FISH to assay large numbers of genes in large areas that will be required for human embryos. And as we start doing that, we begin to realize, and I'm actually amazed at the extremely intricate structure of every little piece of the human embryo. Here's the eye development showing the future retina, the cornea, the lens, and all the connective tissue around it, really developing, and all these cells are talking to each other and forming the shape of the eye and the specific cell types in the eye, and it's really an amazing piece of development. And of course, this is ongoing everywhere in the embryo at the same time. So we need larger scale spatial studies for sure. So what's missing? I think we have already great coverage of many organs, but there are definitely many tissues still not accounted for. So there's a great opportunity for more work focusing on those tissues that have been relatively neglected. We need much higher scale, larger scale and resolution, both in cell numbers and in terms of spatial resolution, and I think in particular temporal resolution. Development happens quite quickly, especially early on, so we need to sample densely. And some things that we haven't touched on so much, but I think are going to be increasingly important, are investigating human diversity, as well as human origins. And I think non-human primates could be very important there, in particular for brain development, because a lot of work has been already done in non-human primates in the past. And as I said, we need a greater emphasis on spatial organization, and we need ways of extracting true lineage and cell trajectory information so that we can query about the cellular ancestry of adult cell types and really trace them back to the embryo. That would be sort of the ultimate goal of this work. And then the time windows, as I said, we're missing the first 3 weeks, the third trimester. We're at the moment mostly missing pediatric and adolescent time points, but we have a whole separate section focusing on that. So with that, I will hand over to Deanne.
Thanks for the introduction. Sarah and Aviv gave excellent introductions to a lot of the data sets and ongoing work. So this gives me the opportunity to talk about pediatric cell atlasing from the perspective of pediatrics itself. So this gives me the opportunity to talk about pediatric atlasing from the pediatrics aspect itself. And essentially, this is from our paper that the reference is below, but essentially we're interested in the interplay between differentiation, the origins of pediatric disease, the functional perturbations that distinguish pediatric disease, and those effects during development. And there are, we are not sure, for example, why pediatric organs can both function and grow and develop at the same time. And that may be, for example, the origins of the unique and, or at least the special and specialized pediatric physiology that we find in pediatric medicine. So we believe that the developmental cell atlas and the pediatric cell atlas can work together on these questions and actually contribute to pediatric research towards children's health. So considering pediatric health from the health point of view, and looking at disease, we believe that the pediatric atlas will actually provide mechanistic insights into childhood health and disease, especially by revealing the cell regulatory mechanisms behind healthy growth and development. And we believe this will have profound implications for research into aging, regenerative medicine, and actual pediatric medicine and treatment, especially those the questions that are cycled around common and rare diseases, including rare genetic diseases and also pediatric cancer. So these are all, there are many areas that are really unique for pediatrics that if people are more familiar with adult medicine may not have an idea of some of the specialized areas of pediatric medicine and where those questions are. So engaging pediatrics researchers will be important in producing an atlas to understand what are the pain points for pediatric research, and especially where the pain points are for keeping children healthy and out of hospital. So we have a holistic and integrated view of a pediatric cell atlas, and I'm not going to go into too much detail about how each of these circles will be represented in the atlas right now. But developmental atlases will clearly be able to give us lineage-specific regulatory mechanisms governing cell differentiation, as some of the slides that Sarah showed would do. And there are ongoing and growing projects, and I know I'm not mentioning everybody here, and I apologize if I've left people out, but there's LungMap and HubMap are actively working on pediatric tissues. The DG-Tex, which is going to be a funded project soon, is going to be taking post-mortem tissues from up to 120 different children across probably at least 30 different organs. And that should allow us to query organ level development, structure, function, and physiology at the pediatric level across 4 different age ranges. Mechanistic insight into disease genes can come from both pediatric genomics and genetics projects such as Kids First, coupled with single cell data, and also computational modeling groups that can take all the data produced from the pediatric cell atlas and actually give us insight into how disease genes impact the lineage networks. And of course, as also Sarah showed, the translational research and organoid atlasing will be very important to identify treatments and from cell type, stage, and mechanisms. And ultimately, we'd like to come up with a roadmap of how all processes define specific pediatric physiology and health and disease. So just pretty much the last slide I have here is that we have a community. Pediatric medicine and pediatric research in general is a very community-driven project. There are a lot of specialists and there's a lot of subspecialties, and people work together very closely on questions in children's health. We have parents involved. We have our physician scientists. We have ethicists and counselors for pediatric and children's concerns. We have computational researchers, our practicing physicians, who are very important to identifying the problems and the problem lists that they're facing. We have our rare disease communities and our biobanking communities that are going to be very important for supporting these research endeavors. We have our technology developers that can help actually discover new ways to sample our lineage-specific programs. We have our consortiums and organizers that can help bring in more of the communities, our funding organizations, which obviously support all this work, and translational researchers that can reach towards treatments for the diseases I just talked about. So even though we're working on a normal cell atlas, or at least a healthy cell atlas to put it better, those healthy cell atlases will help support ultimately pediatric medicine and the questions in pediatric medicine. So that is my, I'll give up my slides to our next speaker. Thank you.
Thank you. Thank you very much, Sten and Deanne.